Greetings, humans. The stream will start soon.
Is it
Um, it takes a bit to.
on? Oh shit, we're there. Tap
Yep.
to unmute.
Yeah.
Hello.
Hello.
You didn't press start on the stream. Okay. Yeah. Margaret, do you want to say something so that I can check I can hear you?
Greetings, humans. Welcome to the Kelvin Club talk. It will start soon, but not yet, because like it's not supposed to start yet.
Said it just takes a bit.
What you got?
The audio works. It's very quiet, but it works. How did we charge it last time?
We charged it from the back, but we didn't use mics, so that's
Seb, how do I turn up the volume on the stream? On the left device, on the leftmost two sliders, up and down. The
Have no
leftmost two
fear.
sliders on the leftmost device. Yeah. Can you say something into the mic? Sorry? Can you say something into the mic? Hello, hello, testing, testing. Hello, testing, testing. Hello, 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 testing, testing. Hello, hello, testing, testing.
I might have just unplugged something.
Don't unplug anything because it usually tends to mess things up.
Yeah, but this will solve it. Now you can charge it up.
Thanks. Okay, so this is a bit of a hazard, so
Hello. Doo-doo-doo. Life is fine. Everything is gonna be okay.
could you actually bring a new glass of water for Virginia for when she's talking? Okay, bare essentials are working now. That's good. Up is more volume. I'll just blabble in here for a bit. Yeah, I think it's a bit loud, at least here-wise. Or? Yeah, but also just for the people who are sitting here, it seems a bit loud, or what do you think? So like if I continue speaking, function in vivo shaped. I think that controls the speaker volume. Stream volume, I'm not as sure how that works. Try what again? Also, actually, Toby, streaming-wise, they can turn on the volume on their laptop, so that's fine. So as long as it's good volume for in the actual room. Yeah. So I'll continue chatting. In the context of IL-10 inhibition, checkpoint blockade. Um, yeah, yeah, I don't know what to do about that. Metabolic manipulation. Yep, I can continue talking. Seems a bit quiet now, or I don't know, maybe it's fine. Yeah. I think that's better. Sure, I'll continue blabbering. I mean, I can always hear myself, but yeah, I can hear it. It's the volume-wise, it seems decent. There is a slight increase, I think.
Okay, so I've fixed the screen volume. I've figured out which button that is.
Okay.
Can you just say something now?
Yep, I'll continue talking. So went to Rockefeller University, began investigating the role of gut microbiota and T-cell related immunity, intestinal barrier function. On T-cell function, gut microbiota, and of intestinal barrier on T-cell function. Okay.
How do you think that is in terms of volume? Because the speakers in the actual room that I'm trying to turn off, basically, and people just turn on, and there's two different buttons which seem to be on all the.
As long as people can hear it here, and
How
yeah.
much in terms of volume do you want, more or less?
I don't know. It's not important what I can hear from here. It's more how it sounded there.
I mean, I think you'll hear it either way. I'm probably just going to press it. Actually, if I sit here, do you want to just say something?
Yeah, I'll continue chatting. So gut microbes and microbial metabolites affect resistance to infection.
I think that's good.
Local, okay.
Hurting my ears sitting right next to the speaker.
Okay,
So
good.
that's probably a decent volume, unless you heard really loud, in which case I'll turn it down.
Okay. Now I'm just going to practice my speech.
In terms of lighting, is there like buttons for lights? What buttons for lights before actually?
Currently I have to focus on my speech because
Okay, I'll tell you. cheese. I am once again cheesing.
Microphones, but we managed to get this thing to work.
Oh, it's messing up. It's doing weird things.
Oh wow.
On the slides. It might be better if I can plug mine in.
Sure.
Yeah.
We might need to change laptops. Apparently, slides are weird.
Yeah, some of the things, the graphics are coming out funny.
Oh, what's happening to the slides?
So here, the arrows are coming out
Is that
not
only,
on top of
is
the.
that only issue because
Let's see.
Essentially, what's happening here is that I just pressed on the link,
Oh yeah, that's weird.
and that just brought me there.
Sometimes it's but sometimes it's a
Is it possible to,
Mac
like-
to PC problem.
Oh,
That's
okay.
usually the problem.
did you download it at all?
Yeah.
Yeah, he downloaded it.
But is it possible to download it as rather than having it as an online file?
You should be able to download the PowerPoint,
Yeah. Oh,
that
you
might
mean
not-
download it here?
but that might not solve the problem. Is
Yeah,
what I'm
yeah.
telling you. Like, unfortunately, this happens at conferences and
Do
stuff
you
all the
have
time.
your laptop?
I do.
Then we'll try see if we can connect that.
This is my audience. All right, it's gonna be like 10 people. Let's see if this fixes it.
Do you also have a USB?
I just have a USB-C connection.
The live stream.
Yeah, because we're currently.
Oh, yeah.
But the live stream's on your laptop.
Oh, is that the
Yeah,
power?
we'll have to log into
On my.
the YouTube.
Oh, it's live streaming?
Yeah.
Oh, I didn't know that.
Oh,
Okay.
sorry.
It's okay. Like, so there would be a bigger audience than I think.
If anyone's actually watching.
Usually we live stream primarily because there are some fellows who are like interested in like seeing from abroad, and because our sponsors from the US. so
Oh,
they can-
cool.
Is it possible that I log into my YouTube account?
Yeah, sure.
Only issue is I don't know what Mac is going on.
Okay.
What is a Mac?
No worries. Let me just find my cursor for one. First of all, is it over there? No. There it is. There it is. Okay, so first let me get on. Oh, you don't have a university connection here. Forget my room. You don't have a Uni of Cam Wi-Fi? There should be.
I barely log on to Wi-Fi on my own home.
Are you serious?
Sometimes.
This is tragically third world is what we're talking about here.
Yeah.
So I don't know how you're going to log on to YouTube then. That was the only thing.
I mean, can we live with the arrows?
So it's not just the arrows, some other things, but I mean whatever. It's a slide.
Okay.
I can live with it if it's
So do we go back to this?
yeah, because otherwise we can't stream it, I guess.
Yeah.
Okay. And then if anything goes wrong when I talk, I'll just blame it on the slides being weird.
Yeah.
It's my scapegoat.
Means excuse.
Whoops, sorry.
So yeah, sorry for the issues with
We
the.
just position the mic so it's easier.
Okay, yeah.
Let me just run through real quick and see what else is funky so I know that what's coming. Look at lips. No, this one's weird. That's okay. You guys don't need to see species names. It's fine.
Oh, so the species names are lost.
Yeah, that's fine. It's not like you guys are really gonna, I mean, I'm guessing you're not gonna say, wait a minute. I need to know the third most abundant butyrate producing species in humans. So hopefully it'll be fine.
We checked previously.
Okay, it's fine. It's a little wonky, but it's okay. Okay,
Okay.
should be fine. So when are we meant to start? Now? Okay.
Yeah, I'll sort of welcome to the Calvin Club's last talk of the term. Today we have Professor Virginia Pedicord, who'll be talking on how the gut microbiome affects things like the immune system, and in some cases even neuronal relationship between neuronal and microbiota systems. She did a PhD at Cornell University, where she then worked on CD8 plus T cells, working on investigating how if you perturb it in various ways, remove various immune checkpoints and alter and for example inhibits IL-10, how that affects the T cell differentiation and function. She then went on to do a postdoc at the Rockefeller Institute, which then started investigating more in the relationship between the gut microbiome and the actual intestinal barrier and and the immune system response there. Finally, she's now working as a PI at the Cambridge Department of Medicine and Cambridge Institute of Therapeutic Immunology and Infectious Disease as a PI, and linking this work additionally to the effects on the immune system as a whole and on the effects, the links between the neuronal system and the gut microbiome. So yeah, as usual, we will generally leave questions to the end of the talk unless
I'm happy to have questions whenever. So if you feel free to interrupt if you'd like, if you have a question about something.
And then, as usual, at the end we'll have drinks and wine. We'll continue, and then you'll have a chance to talk to the speakers. We'll turn out the lights once the talk starts.
Not too dim though, because everyone will fall asleep. Hi everybody. It's actually really nice to have just a small, cozy group. Can I just maybe quickly ask, how many people here are interested in biology and or studying biology or something tangentially related to biomedical research? Okay, good, a couple. Okay, just so I know at what level to kind of introduce things. So that's really great. So yes, so I'm Virginia Pedicord. You can tell from my accent that I'm American. I'm not English. So apologies if I say things incorrectly at any given time. I'm just going to kind of try to give you an overview of what we're doing to try to translate between the mouse microbiota and the human microbiota, and hopefully try to give you an overview of why that matters and why we would even want to do that, because who would want to do that, right? Thank you, Sebastian, for the introduction and for the invitation to come. So yeah, again, just a little of my background and how I came to be interested in this. I was a straight T-cell immunologist during my PhD, and I was studying different areas of activation of T-cells, which are one of your white blood cell types that are important for adaptive immunity, and looking at how those were affected by things like checkpoint blockades, anti-CTLA-4 in particular, which is one of the immunotherapies that is now currently widely used in cancer and got the Nobel Prize a couple of years ago. So that's what I started doing during my PhD. And during my PhD, Giorgio Trincheri from the NIH came and gave a talk at Memorial Sloan Kettering where I was doing my PhD. And he and Romina Goldschmied at the NIH had made this strange observation that in mice that were treated with antibiotics prior to the experiment, when they treated them with chemotherapeutics against cancer that were known to be immunomodulatory, so they have some chemotherapies at the time that they knew modified the immune system. But if they had given the mice antibiotics prior to the experiments didn't work anymore. The chemotherapies didn't work anymore. And this was particularly true for, again, ones that modulated the immune system like oxaliplatin. And at the time, this was kind of just a gee whiz observation of like, well, what is it that these antibiotics are doing? Who is it that the antibiotics are killing that's causing this change in the efficacy of this therapy? And of course, I and a bunch of other people had, a light bulb go off and said, well, it must be something about the endogenous microbes that are being killed off that's modifying this immune response to cancer immunotherapy. I had zero background in working on anything bacteriology or microbiology, but I was very interested to go on and figure out how to study the endogenous microbiota. So for my postdoctoral training, I went across the street to Rockefeller University and started working with Howard Hang and Danielle Musita on gut microbiota and how that affected susceptibility to enteric infections, so intestinal infections. Turns out that had nothing to do with T cells, so you just chase the data. And it was the intestinal epithelial cells themselves that were getting activated by certain enzymes. And we published on that. So we found that certain enzymes were able to actually activate the normal epithelial response and protect against infections. So when I came to Cambridge, I wanted to combine all of these things, basically, and kind of marry my interest in T cells and T cell activation with my kind of new interest in the gut microbiota and intestinal epithelial cells, and see basically how this interaction of this ecosystem of microbes in our gut with the intestinal epithelium and the intestinal immune system kind of led to a lot of other downstream events in host physiology. Now, it turns out, I'll just give step back and give an overview of the gut microbiota for anyone who hasn't been reading about it on Facebook and tons of pop culture stuff. I'm sure everybody has heard about how microbe A or microbe X is associated with susceptibility to pick your favorite disease. But this is what we know now about the human gut microbiota. So overall, we've characterized or identified about 4,600 species of bacteria from the human gut, about 30 species of archaea, 140,000 viruses if we include phages, and about 700 fungal species. This, of course, is not present in every individual. So at any given time, the average individual has somewhere around 1,000 to 2,000 bacterial species in their gut, maybe 3 or 4 archaea species, about 2,000 viruses, and maybe anywhere between 5 and 15 fungal species. So this can really highly vary from individual, and of course, maybe not surprisingly, based on geographic location, diet, all of these things can be really very different between people. This, I think, is maybe not so different from probably what most of us studied in secondary school biology of normal ecosystems, right? There's a diverse number of plants, animals, birds that you have in any given ecosystem contributing different aspects of the function of the overall ecosystem. So I kind of equate this to different people's guts being different ecosystems. So obviously on one end of the spectrum you have things like tropical rainforests, and I put here as an example the Congo River Basin rainforest because it's the second largest rainforest. So in the Congo River Basin rainforest, there are 10,000 types of plants, 600 types of trees, 1,000 bird species, 280 reptile species, 400 mammal species, and this, of course, contributes a huge amount to its host, which is the Earth, our planet, its host's ability to deal with carbon. So for instance, in a very recent study from a group in Leeds, they found that just the peatlands in the central Congo Basin store between 26 and 32 billion tons of carbon. Now, of course, there's other ecosystems even closer by. So the UK ecosystem, if we consider all of the UK, including large forest areas like Galloway Forest in Scotland, the total biodiversity of the UK, by comparison, includes about 1,400 types of plants, 267 bird species, 5 reptile species, and 81 mammal species. But importantly, that ecosystem is still contributing to carbon sequestration, right? So it's approximated that the carbon sequestered by all of the UK in terms of biomass is about 10 million tons of carbon dioxide per year. And I'll come back to this idea of different ecosystems doing similar things. So the human gut microbiota as an ecosystem also I like to think of as a highly dynamic organ as well, because it is highly integrated into its host, into our bodies. In the case of the human gut microbiota, it contains about 30 to 40 trillion microbial cells, and that puts it at about a 1-to-1 ratio with human cells, its host cells, and about 10 to 100 times as many genes as are present in the human. It's highly dynamic, and that unlike our normal body cells, it can change in response to age, diet, different environmental factors, antibiotics, and other kinds of drug uses. So all of these things can, of course, change the members of that ecosystem and the outputs and potential effects on the host that are happening from that ecosystem. So just to introduce you to a few of the ways that we study the microbiota. So, I think it's obviously been known for centuries that we have bugs in our gut and microbes there. But it's only recently that we've started to appreciate the diversity that's there. So I still hear a lot of people saying, oh, most of the bugs in our gut are E. coli, right? Which is actually not true. It was just one of the first bugs that was cultured from our guts. But there's, like I said, thousands of species in there. So with the advent of next-gen sequencing, we actually started to have ways where we could actually see the presence of all of these bugs that we hadn't cultured yet. So the first couple of ways are all sequencing-based. The first is 16S ribosomal RNA gene amplicon sequencing, metagenomic sequencing. Of course, still culturing commensal microbes. It's still the gold standard. We also now use germ-free animals, so animals that are raised in little plastic bubbles, isolators, that allow us to have animals that are completely free of any endogenous microbes. And then we can, of course, give antibiotics, broad-spectrum antibiotics. And then we can take any microbes of interest and introduce them into those germ-free animals or antibiotic-treated animals to study causation if we can. Background on how we do sequencing. So we get fecal samples or rectal swabs, and then we can suspend this in a lysis buffer, extract the DNA, and then for 16S ribosomal RNA amplicon sequencing, we have primers that can either amplify a specific region of the 16S gene, or we can amplify the entire gene. Send that, sequence it, and then what we get out, of course, is a jumble of G, C, Ts, and As, but we can align those to reference genomes and reference 16S sequences to kind of align that to certain taxa. And these can be very specific, but in terms of 16S, of course, there is kind of just broad, a whole genus will have the same 16S sequence, or in some cases, a whole family will have the same 16S sequence. So that comes to the pros and cons of using 16S ribosomal RNA amplicon sequencing. So one of the pros, of course, is that it's inexpensive. It's just a PCR for anybody that has done a PCR. It's pretty straightforward. It's relatively simple to analyze because, again, you're just looking at a relatively short sequence and aligning it and seeing whether it's the same or different from reference genomes. You can classify down to sometimes a species level if the 16S is very unique, but often at best to a genus level, which is still pretty good. And what this does overall, though, is give you a general snapshot of who's there. So if you have a fecal sample from a patient and you want to generally know, what is the community structure there, who is present at a genus level at least, and what is the level of diversity, you can get this picture just from doing this PCR-based sequencing method. The cons are that it is error-prone in terms of classification. So like I said, if that sequence isn't super specific, then you may misclassify what you have there. Beyond having that 16S gene, there's no genetic information. So you don't know what else is in that particular bacterium's genome. Yes, Sebastian?
I was wondering, but wouldn't the bacteria in your gut be selected for or try to actually stay within the gut and try to not end up as part of the fecal samples?
So no, they don't really think about these things. But that's a good question. So there are, I would say primarily no, because a lot of it just comes out. They do stay in the gut lumen for their period of time. Their lifespans are much shorter, of course. But there are bacteria that are very mucosally associated. And those bacteria, you do have to, they're going to come out in a much lower abundance in the feces than they might exist actually in the mucus. So that is a good point. And that's something in animal models we can actually look at, because we can go in and take mucus scrapings, or if we're lucky enough to get some sort of intestinal biopsy, then we can look at those. But yeah, it's a good question. Yeah. Because this is the one that, sorry, so this is a very good question. So this is something that is present in all bacteria, so it's conserved. So we know that we'll get all the bacteria there if we get the 16S gene, but it's highly variable between bacteria in terms of its actual sequence. So if we, for instance, looked for, I don't know if you guys have studied bacterial pathogens too much, if we were looking for genes that are associated with a type 3 secretion system from Salmonella, we would only get Salmonella and some related species out of there. So this is something that we know all bacteria have it, and so it'll give us a profile of all the bacteria. We won't miss them. And yet it's variable enough that we can kind of try to fingerprint the different bacteria based on it. Does that make sense? Good questions. So this is, for maybe not surprising reasons, 16S is how most studies look at the gut microbiota. It's cheap, it's easy, it's relatively easy to analyze. But like I said, beyond the 16S gene, which is what you're amplifying, you're kind of throwing away the rest of the DNA. So you don't see what else is in that particular bacterium's genome, and you don't know what it can do other than who it is. So it's like a name tag. You get the name tag, and you get nothing else about that particular bacteria. What we do is metagenomic sequencing. So it starts out with the same thing. You get a fecal sample, you do DNA extraction, and then this time, though, you just take all of that DNA, break it up into fragments, sequence all of it, and then what we have is usually 150 base pair fragments that we sequence, and we look for the overlap. So that's how we make a longer, what we call a contig. So we assemble those pieces into contigs, and then if you're really lucky and you have a lot of sequencing depth, then you end up with a really long contig. You bin it, and you can make basically the whole genome of a bacterium, or lots of genomes of a lot of bacteria. So this is obviously great because you can get classification down to a strain level. You can actually see differences in 2 different strains of the same species. You get the full phylogenetic structure because you can see the degree of relatedness or not at a genome level. Sorry. Still getting over a cold. You also get genetic information, so you know what's in that genome, what genes they have, and if those genes are annotated, what metabolic functions they might be able to perform. And that gives you your potential functional information. The cons are it's much more expensive, not surprisingly. It's much more specialized to be able to analyze the data, and it's much more computationally heavy. So you can't just do it on your laptop like you can with 16S. I remember being able to just analyze 16S data on my laptop with like an Excel file, and it was super easy. And you can't do that with metagenomic data, especially once you get to hundreds of millions of reads per sample. So why does any of this matter? Why would we want to study the microbiota to begin with, right? Obviously, you know why I started studying it, because I was just curious why it was affecting immunotherapy efficacy. But for everybody else, I'm sure if anyone that looks at any biomedical journal has seen a million papers at this point, probably even in pop media, again, you've heard about some microbe being associated with disease or health, like, oh, okay, if you just have this microbe, then suddenly you're not going to age, and your hair is going to grow beautifully, and you're going to have perfect skin all of a sudden. But so what this made for next-gen sequencing and 16S has allowed for basically everyone to find all kinds of correlations with their favorite disease of pick your favorite microbe. And you'll see here, can I get the pointer? There you go. No, that's not gonna work. I won't do that. Okay. You see cursor? Okay. Yeah, but what you'll see, so it makes for really cool, pretty graphs. They're very colorful, nice correlations and ordination plots. But what you'll see with a lot of these papers, again, is this word correlation, correlation, correlation. So this bug correlates with this disease, an overabundance, underabundance, whatever it is. But there's obviously been a lot of obstacles to taking that beyond correlation and proving causation. So we can see that, a certain bacterial species overabundance is associated with a disease susceptibility. But is that bug actually doing anything to cause or promote that disease happening? And a lot of people don't do that. So the mechanistic studies have been few and far between, and there's reasons for that. It's because it's difficult. So in vitro mechanistic studies, so just doing stuff in cell lines in a dish, obviously don't really capture the complexity of the interactions that are happening in the gut, right? You have a whole community of microbes interacting with each other, feeding each other, competing with each other. Those are interacting with epithelial cells, the underlying immune cells. So in the gut is your largest population of lymphocytes in the body. So all of these guys are all interacting, and it's really hard to recapitulate that in a Petri dish. You can also do in vivo mechanistic studies, but those require animal models unless you have humans that are volunteering to get poked and prodded and have a bunch of random things put into their body. So the most common animal models are laboratory mice because they're easy, they're small, they're easy to house, and they breed really well. But there have been huge obstacles to doing these kind of causative and mechanistic studies in mice because a lot of, quite honestly, grant funders, reviewers, everything else say, well, the human microbiota is very different from the mouse microbiota, and they're absolutely correct. The problem with doing anything directly in mice was that versus humans, where there's been a lot of sequencing done and a lot of culturing done of the human gut microbes, very little has been done or had been done prior to our work on the mouse microbiota. So everyone just kind of ignored it and did their experiments in lab mice, sometimes saw that the experiments didn't repeat between labs and facilities and institutes and kind of just dismissed it as, okay, well, it's different people doing the experiments in different hands. One of the solutions that was proposed early on for this is, oh, well, we have germ-free mice. Let's just take these human microbes and put them into the germ-free mice and examine it that way. The problem with that is that a lot of human gut microbes don't colonize the mouse well, and or they don't do the same things once they're in a mouse gut. So you might introduce 10 microbes from the human gut microbiota into a germ-free mouse. Only 7 of them will colonize if you're lucky. And once they're there, those 7 microbes might not be doing the same thing in that community that they would do in that community of 10 in the human gut. And like I've mentioned, there's very little taxonomic overlap between the mouse and human gut microbiota. So I'm just going to skip forward to this to show you what I mean by that. So if we look, these are just pan-genomes that we made from the human gut microbiota and the mouse gut microbiota. So as you can see here from this very colorful Venn diagram, if we look at the 3,000-ish human genomes that we have here and the 900 only from mouse, we only have about 100 that overlap. So chances are, if you find a bug of interest that's associated with your favorite disease in humans, you won't find that bug in the mouse microbiota and you won't be able to investigate it in that laboratory mouse. And vice versa. If you find a phenotype in the mouse and you see that this interesting new bug is associated with that phenotype, chances are you won't be able to see if it's also associated with that disease or phenotype in humans. So for obvious reasons, a lot of people said, well, we shouldn't be doing mouse work for microbiota research because it doesn't make sense. So what we first did in the lab, which wasn't really what we intended to do starting out, but what we first felt we had to do was overcome some of these obstacles. So the first thing we did was try to tackle this lack of cultured microbes from the laboratory mouse. So what we did is just with Trevor Lawley's lab at the Wellcome Sanger Institute, excuse me, we just high throughput cultured a bunch of bugs from the laboratory mouse. So just normal wild type, normally housed laboratory mice, take their poops, culture them in anaerobic conditions with different culture media, different nutrients. And what we got out of it was 288 isolates, representing about 132 species, 67 genera, 25 families, 6 phyla, 62 of which were completely novel and not been cultured from the mouse before. But what this gave us was a huge new resource. So now we have individual cultured representatives. We can put them back into a germ-free mouse. We can test their supernatants and metabolites in vitro. We can do things with these to actually investigate their function. So what we think we did with this is kind of answer part of the first problem and maybe half of the second problem. So now we can investigate these cultured isolates, and what we can hopefully do is if we can figure out what these mouse commensals are doing, then we can just look for the equivalent bug in humans, right? Easy enough. Not so easy, but. So again, this is our taxonomic overlap. This didn't make sense to me when I first saw this. I mean, I knew it was true because we've looked at their genomes and everything else, but it doesn't make sense to me for the human gut microbiota to be 97% different from the mouse microbiota because we know that they do a lot of the same things. Yes, Basham.
I was wondering, could there be, because things like, presumably our diet is very different from that any other animal, because if there could be a mouse, like some other omnivore, then they would both be eating like stuff that they wouldn't be eating cooked food in the same way. So do we find the same difference between other species?
Yes. So actually, it's very interesting when we look at the excuse me, microbiota of rats, cows, which are ruminants, of course, they're different, fish, everything else. So the microbiota seems to be very species specific. So human to human, it'll be a lot more alike, but human to another species, any other species, even primates, it's actually very species specific. Even if we were to feed the mice McDonald's and everything else, they actually still primarily have their own microbiota. And even wild mice, so there's a big deal made about wild mice being different from laboratory mice, but even wild mice are much, much more like laboratory mice than they are like any other species, or the microbes from wild mice are much more like they are than they are from any other host. And you know, I can infer a lot of reasons of why this might be, but you know, evolution and coevolution and all of these things, but yeah, so that seems to be the case, that the microbiota seems to be very host specific, species specific. But yes. You don't, and that's a really good question. So you don't know, and you have to look at that always. So you always have to follow up on whether it's doing the same thing and whether it's at the same abundance in each of the microbiota. So yeah, exactly. So just to find the equivalence, it didn't make sense to me, like I said, that this would be so different because, like Sebastian said, we're both omnivores. Yes, we have different diets, but at a general level, we have similar diets. We have a protein component, we have a fiber component, we have a carbohydrate component to our diets, right? So it doesn't make sense because the microbiotas, you know, in terms of their function for their host, they're doing a lot of the same things, right? They're fermenting dietary fiber, they're synthesizing vitamins, they're helping us digest or process different kinds of sugars. And they're doing that in humans and they're doing it in mice. We know that they're doing those things in mice. So it doesn't make sense that there should be such a difference. So what we did is we looked on a functional level. And indeed, when we look on a functional level, so which genes they have, which metabolic functions they have. So Interpro, using both Interpro, which is a protein family database, and KEGG, which is a metabolic/gene database, we see that there's actually up to 90% overlap in function between the human gut microbiota and the mouse gut microbiota. So what this tells us is, in one situation, we have Bob doing something, and in another situation, we have Susan doing the same thing. They have different names, and all we have to do is figure out which two are doing the same thing, right? And then we can actually go about investigating the functions in either host. Well, that was a lot harder to tackle at the onset than you would expect because, again, a lot of these tools hadn't been made for mice yet. So we didn't have a really good picture of who was in the mouse gut microbiota and what the names of all of those bacteria were, what was in their genomes, and what their genes were. So this was something that we tackled via a very talented PhD student in my lab who just defended his thesis, Ben Beresford-Jones. And so what he did is he mined through basically all of the publicly deposited metagenomes from all the laboratory studies in the world, and compiled this database of over 26,000 high-quality genomes. So what we did is we took these metagenomes, which is just, again, the sequencing of everything, aligned all of these, assembled the contigs, binned the contigs, and put them into what we call MAGs, so they're metagenome assembled genomes. So they're synthesized genomes based on these overlaps in sequence. And from that, again, he got over 26,000 genomes. This was represents about 1,100 different species of bacteria in laboratory mice from 61 institutes in, I think, 15 countries around the world that had deposited their sequencing data. What does this mean? What does this allow us to do? So other than giving us, again, pretty plots. So one of the problems with mouse metagenomic data before was that everybody was using human databases to classify mouse metagenomic data. As you can probably guess from the Venn diagrams, that doesn't really make much sense. And what ended up happening is you would have a lot of misclassified or unclassified reads because it wouldn't align with anything from the human database. What this first and foremost allows us to do is now we have much better read classification. So now we can actually classify the sequences that we're getting out from mouse metagenomic data and see what species or strain it might align to. And as you can see here, so these are the human databases. So with the human databases, you get on average between 20 and 25% read classification. That means 75% of your reads are getting thrown out as unclassified. And now with our new database, we have upwards of 95% read classification. What this also gave us is a snapshot of where the variation is happening in the global gut microbiota of laboratory mice. So one of the things that vexes biomedical researchers is this irreproducibility. So when a study is done and then you try to do it in your lab and for whatever reason it's not working in your mice. Well, not surprisingly, the biggest source of variation in the gut microbiota of mice is the institute. So every institute seems to have its own microbiota, and this can, as we know, highly affect phenotypes and aspects of physiology, and a lot of times straight up the readouts that people are investigating. So then the next step from that was, again, to try to get some sort of functional annotation so that we could actually figure out where the equivalents were. In order to do this, we had to generate pangenomes. So basically, for every species, if we had, oh, it's just got a mind of its own. For every species, if we had, maybe 6 genomes from that species or 20 genomes from that species, we take all of the genes that exist in all of those genomes and make one long pangenome. So that's basically all of the genes that can be present in that species. It may not be present in every strain from that species, but it's all the genes that can be in that species. And then we functionally annotated those pangenomes, again, with InterProScan and eggnog, so that we can see, of the genes that are functionally annotated, what can they do. What this allowed us to do is then do the same thing with the human microbiota database, and then we can find who is the most functionally equivalent between the mouse microbiota and the human microbiota. So if there are 40 functions that you're looking at, it's never that simple, but if there's 40 functions you're looking at and these two microbes have 39 of the same functions in common, those will be very closely related, whereas these two other microbes that only have 7 in common will be functionally not as closely related. What's really interesting and maybe surprising for a lot of people that do phylogenetic analyses, so everyone kind of expects that a microbe that is phylogenetically closely related is going to be doing most of the same things. And by and large, that's mostly true. But what we actually found is there are Additionally, many times where the closest functionally related microbe is actually phylogenetically distant. So it's actually doing a lot of the same functions, but yeah, again, has a completely different name. And that's what these arches mean here. So basically at the different taxonomic levels, these are the closest neighbors. And then you can see, I don't know if you can see the colors as well. But even in the red, even at the genus level, you see that sometimes the closest functional neighbor is not the one that's closest next to it phylogenetically. Why does this matter? So where could we apply this immediately? In humans, it's known that certain drugs are metabolized by resident commensal microbes, and these include glucocorticoids, L-DOPA for Parkinson's disease. So we looked for some of these genes that are present in the human microbiota to see whether they were present in the mouse microbiota and where they were doing the same thing. And as you might expect, you have several different scenarios here. In some cases, you have, in the case of diltiazem and the gene BT496, you have largely the same species performing the same function. So both humans and mice have Bacteroides theta iota micron. That's just fun to say fast. They both have the same species and they're doing the same thing. They both have this gene in mice and humans. In a different situation, you have the same function being performed by different species. So in humans, it's largely Ficicoladorii, and in mice, while they have Ficicoladorii, the species that's actually harboring this gene more often is Ficicola vulgatus. And then, of course, in other cases, a function will exist entirely in one host and not the other, as is the case here, where you only have this function encoded in Klebsiella in humans, but not at all in mice. Something that's probably more broadly relevant, again, one of the important things that the gut microbiota does is ferment dietary fiber into short-chain fatty acids. Short-chain fatty acids have lots and lots of functions in the host. In terms of they provide energy to epithelial cells, but they also signal distantly. They can be HDAC inhibitors, so histone deacetylase inhibitors, change epigenetic modifications in the genome. And one of the short-chain fatty acids that's abundantly produced in the gut is butyrate. And this is only done by the microbiota, so the host doesn't do this, by the way. So there are two main microbial pathways for butyrate synthesis. One is the BCOAT direct pathway, and the other is the indirect pathway, or PTB-BUK pathway. It's known in humans that BCOAT is encoded by very common bacteria like Faecalibacterium proxinetiae and Rosaburia, but it was not really known which bacteria in the mice do this. So we know that there's tons of butyrate in the mouse gut, but nobody knew who was producing that butyrate. And sorry that the fonts are largely messed up in some of this, but in case anybody wants to look at the species names, you can ask me after. But so here's what was interesting, of course, when we look again at this, is the most abundant bacteria in the human gut microbiota that are producing via the BCOAT producing butyrate it's entirely different in the mouse who's producing. It's happening, and there's lots of bacteria there that encode BCOAT, but they're all phylogenetically different from the humans. So proof of concept with what this tool allows us to do is, again, we can go and actually take our isolates of those mouse species, grow them out, and see if they're producing butyrate in the way that we predict they are based on their genomes. And so here in the first 5 are mouse bugs. Again, completely different species from the human ones. So these are the 2 biggest butyrate producers in each pathway from the human gut microbiota, and these are the most abundant butyrate producers in the mouse microbiota. A lot of them have fun names like MGBC 164771 because we don't have a species name for them yet. We have gotten to name a couple now at this point, which is fun. But yeah, so proof of principle is when we predict that they are functionally equivalent, at least in the case of butyrate production, they actually are functionally equivalent. They are producing butyrate like we expect that they are. So I guess kind of the punchline for all of this is what's in a name, not to be too Shakespearean, but like I said, so you're redecorating your house, and in one case you have Phil and Jaime working on digging a pond in the garden, and Susie's working on the walls, and in the other case you have Khalid working on the walls, and Stefan and Anne are working on digging that pond in the garden. At the end of the day, does it really matter what their names are, or does it really matter what they're doing and what their skills are and what their capabilities are, what their tools are? And that's going back to the ecosystem, kind of the same thing. So if we look at the sapele trees that are in the Congo rainforest versus the Sitka spruce, which is in all over England and Scotland, they're all able to sequester carbon, right? Albeit at very different levels, but they're doing the same thing, even if they're in a different place and have a completely different name. So how can we apply all of this to human disease, right? How does this get us to actually investigating something that might be biomedically interesting? Well, not surprisingly, not long after Giorgio Trincheri and Romina started presenting that data on immunotherapy, a lot of other people started looking. And at this point, I can't even count how many papers have come out, again, associating different species with response or non-response to cancer immunotherapies, especially T-cell mediated cancer immunotherapies. But what you might notice if you're actually reading the slides at this point is they all have different names. So everybody's finding a different bug associated with response or non-response to immunotherapy. And the data are quite convincing. So I don't want to say that any of these are wrong, but I think that it might be wrong to be paying attention to the names of the bugs rather than what they're doing. We know that a lot of common microbial metabolites, so things that many bugs in the gut microbiota produce, especially cell wall components from commensal microbes, can modulate immune function. So who here has done any sort of immunology coursework yet? Awesome. Okay, so what you may remember from your immunology course is there are molecular patterns that a lot of pathogens, it's how we studied it to begin with, pathogens encode, that are sensed by what are called pattern recognition receptors in the immune system. And these pattern recognition receptors are on most immune cells, but they're also on things like epithelial cells at any barrier surface. So it's a way for our body to sense the presence of something foreign. And it's important in terms of our immune responses to sense things that are potentially pathogenic or disease-causing, but they actually also alert us to the presence of metabolites from just our commensal microbiota. Not surprising, right? So we know that specific metabolites from the microbiota, or we expect that specific metabolites from the microbiota can modulate immune responses to these checkpoint blockade inhibitors or checkpoint blockades for cancer immunotherapy. A couple of papers have come out looking at some of these specific metabolites. So actually my former lab looking at muripeptides that signal through NOD2 receptors. Also, Kathy McCoy's group in Canada found that inosine is able to signal to the immune system through a previously unknown receptor. So what, just for an example for this, so instead of focusing on, so in the case of, in the case of both labs, so you'll see here, like Howard's lab found Enterococcus, because Enterococcus is one of the species that can produce these peptidoglycan fragments that can signal to the immune system. And Kathy's lab was looking largely at Bifidobacterium and Lactobacillus and their production of inosine. But what we would propose to do is, instead of focusing on the species, focus on the function. So in the case of inosine synthesis, for example, we can look for these genes in the inosine synthesis pathway, agnostic of the species, and look for the abundance of these genes in the microbiota and say, okay, is this microbiome more or less likely to be producing large amounts of inosine, or is it not? And would that make the patient maybe more likely or less likely to respond to cancer immunotherapy? Can we add it back if they don't have very much? And we know ahead of time, can we make them more responsive to immunotherapy? Oh, it's going crazy. One of the main components of the gram-negative bacterial cell wall is lipopolysaccharide, or LPS. This is in, again, lots of bacteria. Actually, one of the most abundant phyla bacteria in the gut are Bacteroides, and these are gram-negative. What we have found, this is a talented postdoc in my lab, Pushpenu Sardar. So what we did is these ordination analyses, basically clustering analyses by all the different parameters of data that we have. When he clustered the data from immunotherapy patient microbiomes, when he clustered the data based on gram-negative species, you see that we actually don't see, so in red is non-responders and blue is responders to immunotherapy. You can actually see if we look by species, there's no real clustering, segregating a responder from a non-responder. However, when we look at the genes involved in cell wall biosynthesis for gram-negative cell walls, we see a really nice segregation. So now we see that responders are clustering over here and non-responders are clustering over here. And one of the genes that's highly different between these two clusters is this gene LPXM. And again, Ben Beresford-Jones looked again through data sets of immunotherapy patients, responders and non-responders, and again found that there was a significant difference in responders and non-responders in that responders to immunotherapy in general had a higher abundance of LPXM encoding bacteria in their microbiomes prior to starting immunotherapy. Now, what is LPXM? So now we can go back and see what is the function of this. So LPXM is kind of a terminal part of the LPS biosynthesis pathway. So you have kind of the first set of LPX genes, CDHK, that produce lipid A, which is part of the LPS portion of the gram-negative bacterial cell wall. And then there's different acyl chains that are added on to lipid A. So LPXL adds on a fifth acyl chain to make pentacylated LPS, and LPXM adds on a sixth acyl chain to make hexacylated LPS. What's important about this, and what was previously known kind of just straight in bacteriology circles, is that these different acyl chains determine how well the pattern recognition receptor, toll-like receptor 4, TLR4, gets activated. So tetra and pentaacylated lipid A from LPS either weakly agonizes or inhibits TLR4 signaling, whereas hexacylated LPS actually gives a very strong signal through TLR4. And what this does is activate inflammatory genes downstream in the cell of interest. So we thought this was really interesting that patients that have this more active form of LPS were responding better to immunotherapy. So we looked to see directly comparing these two types of LPS in vitro. So we took kind of representative. So hexacylated LPS is present in E. coli and most gamma proteobacteria. Pentacylated LPS is present in most Bacteroides, like Porphyromonas gingivalis. So we took purified LPS from each of these, E. coli as our hexacylated and Porphyromonas as our pentacylated, and compared how well they were able to stimulate a reporter cell line of human monocyte macrophages. And as you can see here, so E. coli LPS at just 1 nanogram per mil is able to stimulate at a similar level to the pentacylated Porphyromonas LPS at about 10,000 times that concentration. So the hexacylated LPS is indeed signaling way higher through TLR4, and we've done this in a TLR4 knockout to prove that it is TLR4 mediated. So hopefully I can kind of convince you here. So what this gives us is a new tool to really mechanistically investigate what's going on, translate between what we see in human patients by correlation, take it back to in vitro and in vivo mouse models and see how that's affecting things. And that's what we're doing now. So we basically are translating back into mouse tumor models to see if we can add hexacylated LPS in to mice that lack it to improve tumor immunotherapy responses. And to do that, we can select isolates from our culture collection now and also colonize or cohort colonize germ-free mice. We could also purify the metabolites, the LPS, like we have done, or just the lipid A, and then add that back in vitro or add that specifically to the mice just to see if that itself will improve their responsiveness to immunotherapy. So just to sum up, we have this new database and bioinformatic resources now that allow us to map and figure out who the functional equivalents are, what their names are, between the mouse and the human gut microbiotas. We have a culture collection so that once we find out what that equivalent is in mice, we can put it into germ-free mice or investigate it in vitro. And then we think if we kind of stay agnostic of the names and the taxonomy, it's not that we completely ignore it, but if we primarily focus on the function, that this will actually allow us to get at the mechanisms of the microbiota more easily rather than just finding these correlations between an increase in a bug of name A and a bug of name X between different disease states. So with that, I'll thank everybody from my lab, which is a small lab. We have Amelia, Ben, who again just defended, Pushpendu, Satoshi, and Wangmayu. Everybody at the University Department of Medicine and the Pathology, Department of Pathology with whom we collaborate. Simon Clare, who's helped us a lot with our mouse work. We poached him away from Wellcome Sanger, so now he's back, he's at the university. And then I still collaborate with my good friend at Sloan Kettering in New York to do our metabolomics work. And this is my lab, and I'm happy to take any more questions. Thanks, guys. Some oral bugs that make it through, so we do have some of that, but actually, yeah, it is relatively easy. So Porphyromonas is actually an oral one. It is relatively easy to take samples from the mouth as well. However, there are niches, obviously, so there's some kind of microaerophilic bacteria that will live like up between the gums and the tooth and things like that. So that's harder to sample, those kinds of things. So you don't get really kind of that big of an ecosystem picture from oral microbiota, but there are people studying that. I mean, primarily in things for periodontal disease and that kind of stuff. Yes, yeah, heart disease.
Kind of following on from that, do you think that like other areas that are colonized by bacteria could also have an effect on like the immune system?
Absolutely.
Maybe oral bacteria, so it's understood you can actually have it just
Yeah. Absolutely. So, and we know this. So we already know like skin bacteria can have effects on immune development in skin. It's a newer thing. It's not that new, but people have more recently, maybe more so because of COVID and things like that, started studying more of the respiratory tract microbiota. All of these are less diverse than the gut microbiota for maybe not surprising reasons. So you won't, instead of a community of thousands of bacteria, you'll have a couple of dozen in some areas of the body. But we absolutely do know that, for instance, the skin bacteria are important for Th17 T-cell development, if you've studied that already in your immunology class. So, good question.
What are the current, sorry, can you hear me?
Okay.
What are the current challenges facing your lab or within the field of research that's just starting out?
I actually think that's a good question. There's probably a ton. Funding is always one. No, that's just the eternal problem. But actually, I think in some ways, having this kind of deluge of correlative data over the past decade or so has actually been detrimental in some ways to our field because it's kind of diluted out the interest in the functional investigation. And I think a lot of people kind of lost trust in microbiota research as well because they have this response of like, well, yeah, okay, we see a different bug every week that's associated with this disease and all of this stuff. Clearly they don't know what they're doing and they don't, and no one's really looking at this the right way. So I think, even in my department, quite honestly, there are people that just are very dismissive of it because they think that, that, oh, we're just like reaching at correlations all the time, and that, you know, everybody comes up with a different answer, so how can any of those answers actually be valid? And there's, you know, some validity to those arguments against it. So in a way, it's kind of like we have to fight back against that now to kind of say, actually, we can do functional analyses and we can do mechanistic studies now, and we should be doing that. So but I'm sure there's lots of other things that I complain about on a regular basis. Any other questions? Yeah.
Determine which bacteria produces a certain substance.
So that's a good question. So, I mean, bioinformatically, we do it at a genetic level. So, you know, if, you know, microbe A has BCOAT, then we know it has the enzyme where it can produce butyrate. That doesn't mean that it does, right? And it doesn't mean that it does all the time. So the only way to validate it is to actually put it in the situation of interest, right? So put it into its complex system and then deprive it of fiber, for instance, right? Or put it on a high-fat diet and then see, is that microbe still able to produce what we think it's producing? How does it respond? Does it even survive in a complex ecosystem in that situation? So ultimately, we always have to kind of put it back and then see what it's doing. One of the ways that we do that artificially now is if we just culture it in vitro, you know, with plenty of nutrients and everything that it likes in a nice, warm, cozy environment, is it able to produce it? And usually it does. But whether it will in a complex situation is always a question. We have to actually try to recapitulate that situation if we want to answer the question.
Last question from me. You mentioned, I can't remember what you called them, I think you may have called them germ-free animals.
I meant to put a picture up because it's actually really fun, the bubbles that are in, but yeah.
So I'm not going to ask about how you actually get the germ-free stuff, because that's sort of, particularly for placental mammals, that sounds like a real pain in itself. But
C-section.
well. But what I was going to ask is, once you've got them, I'm sort of half surprised that with such a complicated and diverse microbiome, that they're viable at all. You always imagine that some would be completely dependent, and if you took all of them out, that they wouldn't die. But that sounds like that's not the case, is it?
It's not the case, but you're absolutely correct. They are not normal. And that's something that I think was overlooked for a long time, and is still honestly overlooked to this day. People publish things, you know, recolonizing germ-free mice, and they say that it does this, but we don't see it in SBF, but that's okay. SBF is specific pathogen-free, which just means normal, normally housed, normal microbiota. They're different. So they don't, their immune systems don't develop the same way. Their epithelial cells and the gut don't develop the same way. But they overall are able to function. They do have weirdnesses, so you have to provide them with basically a lot more nutrients because obviously they don't metabolize and absorb the nutrients from their diet as well as they would if they had microbes. Like I said, their immune system is highly underdeveloped, but that seems to be okay because they're not really challenged with anything either. So they do okay with that. But what we were actually, and I think a lot of people were kind of okay with that. It's like, okay, they survive and they seem to be okay as long as we keep them in these bubbles. Of course, anytime you take them out, they're highly susceptible to infection. Incredibly, like for instance, in a normally housed mouse, we could give them 10 million salmonella, CFU of salmonella, and they would maybe get a little bit sick and likely recover. For a germ-free mouse, we can give them 10, and they will die within 4 days. So it's incredible that lack of immune development, how much it is affected. But again, if they stay in the bubble, they're fine. But what we're starting to look at in my lab, which people I think have kind of overlooked, is we do see differences in brain development. So they do seem to function, but given certain insults, they are more susceptible, for instance, to symptoms of major depression. And we think that this can go back to just there are microbial metabolites that make it into the circulation, and that the entire body uses to cue. Sorry?
It's not just that they're in a bubble and they have nothing to play with.
Yeah. Well, they have the same things to play with in their cages that, well, that's a whole other thing. But okay, yeah. It's not We don't have any free-range mice, sadly, but yeah. So they're all in similar kind of housing conditions. Yeah, so we do see like their brains, their central nervous system develops slightly different. Again, it's still functioning at a broad functional level, but there are differences.
Has there been any studies into how that might be related to like depression and so correlated to variations in microbiota?
Sorry, can you say that again? Because I coughed.
Has there been any studies into like the correlation of variation in microbiota and like different disorders, like mental health disorders and stuff like that?
So that's what we hypothesize. So we're working on it right now. Basically the hypothesis would be, because obviously, we don't expect that it's going to be dramatic, but the hypothesis would be that, perhaps exposure to certain metabolites, microbial metabolites during development versus not, means an overdevelopment or underdevelopment of a certain part of the brain or a certain cell type in the brain. And again, the overall function may be fine, but in response to a certain insult, this brain might be more prone to a neuropsychiatric disorder than this brain would be, just because it doesn't have the cell type of interest there or isn't as mature, that cell type, that kind of thing. That's the hypothesis. Right now we're doing it in mice, so they don't obviously have the same exact situations, but we do have a collaboration with a group in Addenbrooke's looking at postoperative delirium and seeing if there's any microbial associations between patients that are susceptible to having postoperative delirium versus ones that don't.
Okay, I'll just ask one last question and then we'll head back to the I was wondering, like, say that like you fed like a mouse and a human the exact same diet, like say where does the differences in where do you get the bacteria from in your gut? And then related to that, does that mean that you then have more with the gut microbiome that you'll have for the rest of your life?
That's a really good question. So it is largely dictated from your family structure and from what you received during birth. Of course, this is really most visibly seen in the difference between natural birth and C-section babies. However, this can and does largely normalize by about the second year of life, and then it becomes largely environmental. So your microbiota will be largely the same as your family or whoever you're living with. But then, of course, it is influenced by diet. But what tends to happen is people you're living with and family structures tend to eat the same things, right? So it's hard to segregate, which of those things are contributing more to changes. But also just over age, your microbiota evolves a little bit. And then again, if you change your living situation, you go off to university, you're eating different things, your microbiota will change a bit then. You get an acute infection, enteric infection, your immune system goes crazy, kills off a bunch of stuff, your microbiota will change then, and it'll take two different people that have the exact same infection or the exact same antibiotics, their microbiotas will recover at different trajectories, just depending on I think, in some ways stochastic reasons and other reasons just diet or whatever else. So lots of variables, yeah.
Once again, thank you very much for coming and talking with us and then moving all into the local wine issues. Thank you very much.
I'll unplug your laptop so you can have it back. That was fun.
Sorry for the technical issues.
Oh, no. Like I said, I'm sure everybody was missing that third bacterial species name, but
Yeah.
it's okay. I find it interesting, but that's why I work on it.
Yeah.
I don't fully expect other people to find it.
Have you been leaning more towards working more on the field, like because there were kind of three different things that, at least in the that were mentioned in your biography. Like one is like linked between gut microbiome and neural. Then there's the like immune relation, and then finally there's the just how does it differ between. Which of those have you been focusing on any of those, or just all of them? Okay, yeah.
Yeah, I've been warned about this. My lab is all over the place, but I guess my philosophy, it's kind of because of the people in my lab. So Satoshi is is a very talented doctor from Japan, and he's a neurobiologist, and he just came to the lab to kind of work on this. And he's like really interested in the gut-brain axis, and I said, I know nothing about neurobiology, but absolutely this is really cool, and you should do what you're good at and what you're interested in, so go for it.
Cool.
And he did. So it really is, I like to let people in the lab kind of chase whatever they're interested in, and that has kind of taken it in very different directions, but I think that's cool. I'm okay with it.
Do you still do like the projects yourself or like in the lab?
I help them, but they kick me out of the lab a lot. So, because I'm like, why is this pipette here? And they're like, oh, okay, thanks, bye. And they're like, just get out. So no, I mostly help them with dissections, and then I help them with some of the mouse work and stuff like that. So, but yeah, other than that, and then we just sit and meet and talk about experiments and like what they want to do and how they should answer it. That's, I think, mostly my job now is curating. Is like they go and they say, I'm wondering about this, and I want to do this experiment. I'm like, okay, will that experiment answer the question that you're trying to ask? Maybe not. Okay, how can we figure out an experiment that will answer the question that you're trying to ask? And then we go through everything, and then it doesn't work. Okay, so what else can we do, right? You know, so that's a lot of what I do. That and writing grants. So, I'm like, my job is to curate your ideas and find money for you to use at this point. Do you need to get your? Oh, good night. Thanks for staying. Sebastian, you want your computer? Do you?
I'll go later on.
Okay.
Yeah.
I have right now postdocs and 2 PhD students. One of them just finished and still trying to finish up more. And then one of my postdocs is also finishing in
laptop? Whose laptop?
Huh? Maybe turn these some of it off. So
Your laptop?